Can everyone see, or can anyone see my main screen?
Yes.
Yes.
Okay. Great. Well, like I said, thank you for logging on. You know, it's interesting times that we're living through, but it's wonderful that we get to keep the scientific enterprise going and have what I hope to be a fun conversation about some of this work in this setting. My talk today is on brain network aging, and I'm going to walk you through some efforts that my lab and myself have been investing our energies towards in terms of understanding how our brain changes as we grow older. And as Dr. Maestri mentioned, the emphasis will be on the science of networks, since we think there's a lot to be learned from studying the brain as a network. Before I begin, I just want to put some context into what motivates some of the work that we pursue. And to do that, I'm going to focus on a few quick images that summarize what I think to be some important and challenging observations that we are trying to understand and that we're trying to anticipate. So the first is that the global population is getting older, right? So if we look at the number of people age 65 and above as a proportion of the population, even by region, we could see that in the next 10 years, the proportion of individuals who are over 65 is going to dramatically increase. And this is, a testament to the quality of healthcare and the quality of medicine and health in general that's been increasing, whereby leading causes of death and illness have shifted from infectious and parasitic diseases to non-communicable diseases. But coupled with this observation that we're getting older, there's also a challenging reality, and that reality is summarized in this figure here. After 65, the incidence of cognitive dysfunction really does increase dramatically, almost exponentially. With every additional 5 years, the proportion of individuals with dementia approximately doubles. Now, this obviously is a figure that highlights age-related illness, but even in the absence of illness, with increasing age, we see changes and differences in cognitive function. Even in the absence of disease, as I said, we see progressive declines in many aspects of cognition, including, so this is a figure that was put together by one of my colleagues here at UT Dallas, Denise Park, and her colleague Patty Roeder-Lorenz. And what they're plotting here are the differences in cognitive ability as a function of age. And you could see multiple different domains of cognitive ability, and this is an unsettling reality as well, right? So we see speed of processing, working memory, and long-term memory all declined even from the age of 20s. So many of us here in this presentation are unfortunately probably on somewhere on this graph. Now, it's not all a bleak story, because many people have also documented that other aspects of cognition seem to show some preservation with increasing age, and that's highlighted here by the world knowledge lines. But the general take home is that, again, even in the absence of any diseases, we tend to see declines in cognitive function. Now, these lines, of course, are a summary of groups of individuals, but there's an interesting nuance to this, and that nuance is depicted in this final figure. There does seem to exist considerable individual variability, and we're probably all familiar and maybe even experience some of this variability and are self-aware of it. Some people seem to age more gracefully than others. So what's being depicted here are actually changes for different individuals in terms of their perceptual speed. It's just one of the domains of cognition that I mentioned earlier. And this is these are multiple individuals who were tested on this set of cognitive tasks up to 6 years. And you can see that any given individual's line can vary from another. Right? So in general, while we see this general trend where increasing age is associated with decreasing ability, different individuals seem to follow a different pattern of change. Some individuals exhibit decline much more rapidly, and others seem to be preserving cognitive function as they grow older.
Dr. Gagan, I have a question for you.
Yes, of course.
Sorry to interrupt you, but
no problem.
some measurement like the last figure that you are showing to us, there are some people who increase this perceptual speed. This is an error for the tools, or really at that age you can increase the speed?
That's a wonderful question. I mean, I think we'd all like to we'd all like to think that there are ways of increasing cognitive ability in older age, and I think that's of course a very large topic of focus. Whether or not there's forms of cognitive training that might enhance abilities. Some of it, however, does come from errors in the measurement tools. So there are things such as practice effects that can permeate some of these data points, where some individuals get better because they've learned the specific tasks that are being tested. I don't know that this specific data depiction was as vulnerable to those practice effects, but some of the increases do sometimes relate to that. Another aspect that is related to measurement error is just the reliability of the measures. So a given measure, if you measure 2 days back to back, might not necessarily exhibit high reliability. So those are always considerations to take into account when you see some of those increases. But again, I think that's a big question as to whether or not some of those increases in some of those individuals are real.
Thank you.
Sure. So I think, when we take a step back and look at these and consider these 4 observations, we're met with some critical questions that we really need to answer. The first is what causes age-accompanied cognitive decline? And can we identify those causes? Of course, if our ultimate goal is to slow down or even halt any age-related decline, we need to understand the underlying causal sources. And the second is, why is it that some people seem to be more vulnerable to decline than others? There's clearly individual variability in terms of normal cognitive aging, but also cognitive dysfunction. And why are some people simply more vulnerable? And I hope to start to answer some of these questions or give you a sense of how we're thinking about these questions in the presentation today. Now, the work on brain aging, of course, is not new. There's been multiple imaging-based measures that have been examined to try to understand the causes of age-related decline and the individuability in that decline. And I'm trying to summarize some of the larger measures that have been used over the years in this slide here. These include task-related activity, so having a subject, a participant in an MRI scanner and doing a cognitive task and looking at bold activation while they're performing that task. But then, of course, there's also been measurement of brain structure, including gray matter structure and white matter structure. And then a whole other class of analyses that have focused on things such as FDG metabolism or neurotransmitter uptake and so forth. And these have all actually yielded promising signals that there are measures that are collectible that might help us elucidate the sources of age-related decline. But it seems to be the case, I think this is a fair statement, that we have yet to establish a clear link between any of these observed age-related brain changes and cognitive decline. So there's still considerable work to be done. Another reality is that there's presently no real reliable measures of brain health prior to the onset of clinical symptoms. It's one of the outlier domains where if you go get your annual checkup at your physician, there isn't really much done in terms of quantitative measurement to understand your trajectory or your potential vulnerability to subsequent decline. So that's something that we still need to work on. So in the context of all those other measures, I want to present to you just our working hypothesis. And again, this has been the hypothesis that we've been testing for the last few years in my lab. The hypothesis is that age-related cognitive decline can be better understood by studying the brain network changes that occur with age. We're trying to use a brain network framework to help us understand some of that individual variability in age-related cognitive decline. So why is it that we think this is a better way of studying brain aging? Well, the first thing to consider is a simple reality that your brain is a network. No neuron or no brain region works in isolation, and we've known this for a long time. When we think about cognition and we try to understand cognition, we understand cognition to be an emergent product of distributed patterns of activity that are across multiple cortical and subcortical structures. So thinking about the brain as a network and its dysfunctions as a dysfunction of the network seems as an appropriate level of analysis. This idea isn't a new one, though. There's been many prominent ideas, theoretical ideas, and applications of those ideas in brain aging research that have invoked a network-related concept. If you're familiar with some of this literature, you'll be some of these terms will be familiar to you, and those include network compensation, dedifferentiation, scaffolds, neural reserve, and disconnection, disintegration. These are all network-related concepts that have tried to understand brain aging or describe brain aging as a problem of networks. And again, I think these have had immense value because they seem to be an appropriate level of analysis at which to start to relate changes in broad domains of cognition that we see with age to changes in information processing. Now, thinking about the brain as a network can be helped by considering the fact that there is a real science of networks. And the analysis of networks can actually be conducted using formal methods. Now, the basic way to think about networks, of course, is that a network consists of things and their interactions. And in the language of networks, we refer to those things as nodes and edges, respectively. Now, networks can be lots of different things. We all interact with networks all the time. Networks can be technological, such as the internet. Networks can be sociological, individuals in a group. In this case, this is a famous social network of a karate club and individuals in a karate club that were part of a single dojo that ultimately got fractured into 2 dojos. And finally, those things can be biological. So protein-protein interactions can be depicted as a network as well. And we think that the science of networks is an appropriate discipline with which to leverage and to understand healthy and pathological aging. We think that healthy and pathological aging might be a network effect, and that we can learn a lot about those processes by leaning on some of the insight that's been gained by examining these other types of networks. So that's what we're trying to do. So how can we study aging brain networks? This isn't as straightforward, it turns out, as simply throwing your data into an app and having it spit out a number. As we started to walk down this path, we realized there was considerable challenges. So here's the first challenge. And that's one of what type of data are available to us. Brain networks exist at multiple spatial scales. And given that we were interested in studying alive behaving humans, we're constrained by the tool we're using. With non-invasive imaging, we can currently study the organization of brain area networks in humans. So if we think about the brain as a network, the first level at which we probably gravitate towards is in the network of neurons. But again, neurons are too small in order for us to image them with non-invasive imaging. We are, however, able to image things on the order of 0.5 to 3 millimeters, 3 cubic millimeters. So that puts us at the level of areas, which are groups of functionally related neurons. But of course, we can also understand systems, which are groups of areas, and so forth. So that's the first pragmatic issue that we're faced with, and that is the scale at which we operationalize our findings and the scale at which we try to make any headway towards understanding brain network aging. In terms of thinking about interactions, there are of course multiple ways of thinking about interactions. The first is thinking about how those different areas interact with one another via yes? Oh. Was there a question? Maybe not.
No, I think it was just a glitch of a computer glitch or something.
Okay. Oh, no worries. I want to ask, can you see my mouse if I'm pointing to things?
Yes, we can.
Oh, great. Okay. That makes it easier. So the first way to think about interactions between areas is to quantify the extent to which they're structurally connected via fiber pathways. And methods such as diffusion imaging allow us to do that. The other way to think about relations between areas is to look at functional relations. So what are the 2 areas doing when a given individual moves their hands in a coordinated fashion? And that would be a functional relationship. I'm going to focus on functional relationships for the purpose of this talk because that's where we've been focusing our efforts. Now, it turns out the functional relationships can be quantified even in the absence of tasks. And some of you are probably familiar with this, since this area of research has actually picked up a lot of momentum in the last few decades. But it turns out that the brain is functionally organized at rest. Even in the absence of task, if you look at functional activity, different sets of brain regions that do similar things are functionally correlated. And I can depict that here with a simple, straightforward example. If we take a region of interest in your motor cortex, here I'm going to take your left motor cortex, and I have someone just sit in the scanner doing nothing, stare at a passive fixation crosshair, and ensure that they're awake, but just have them lay there, and I look at the signal. It'll wax and wane in some seemingly random fashion. But the really surprising thing is that if you took the homotopic region in the right motor cortex, so I'll plot that in yellow, and plotted it, it would look something like this. So it looks surprisingly similar. Now, the first question you might be wondering is, well, maybe the whole brain is just doing this. And sure enough, if I take a control region, it turns out that it's not doing this. So here's the control region, and it's not really doing the same thing. And this reflects a level of organization whereby regions of the brain that are functionally related. Is there another question, or is that a mic? Okay. Regions of the brain that are functionally related seem to exhibit correlated patterns of activity, even at rest, even in the absence of any task. And just to paint the rest of the picture here for you, again, I can take that region of left motor cortex and just plot all the other brain regions that have similar patterns at rest, and you reconstruct a collection of brain regions that are all involved in motor execution and motor preparation. So these initial observations were made by Bartha Biswal and a few other colleagues. But since then, resting state correlation patterns have been described across many brain systems that all mediate distinct domains of cognitive operations. So our focus on understanding how brain areas are related to one another is on analyzing resting state networks.
Dr. Gagan, I have a question for you.
Yes.
Yeah, okay. You're talking about the bold related to the imaging, right?
Bold related to what, sorry, imagine?
Yes.
It's not really imagining. So they're not instructed to do anything, right? They're instructed to just lay in the scanner and passively observe a fixation crosshair. Okay,
but I understand that you are saying that there's symmetry in the pattern that this part of the brain is having, but the signal that you showed before, that they are similar because they are similar structure, is what you're trying to say.
They're functionally similar, yeah. And of course, those functional similarities are mediated by structure. But one of the things we've learned about these resting state correlations is it's not purely structure. So two things need not be directly connected via direct fiber path, and they can still be highly correlated. So left V1 and right V1 are a good example of that. But what we think this is a combination of structural relationships and functional relationships. So it's almost Hebbian. Things that are firing together over your lifetime are going to start correlating even in the absence of task.
Okay, so sorry for the naive question, but I am not a neurologist, so just trying to understand. The voltage, the signal that you are showing us, is electrical signal, or what is that?
This is the blood oxygen level dependent signal, right? So this is just a processed bold time course. So it comes from echoplanar imaging in an MRI scanner that's been outfitted with a head coil, and we're just looking at the ratio of oxygenated to deoxygenated blood.
Okay, good. Thank you very much.
Sure.
We have a question in the chat.
Okay.
Dr. Firat asks, seems like the control region pre-acts other regions. Is this a correct observation?
Sorry, the word was pre-acts?
Yeah.
So proceeds. This is just meant as an example, but that's a really interesting and good question, whether or not there exists lag structure in some of these resting state correlations. And there is some evidence that some regions, maybe not specifically this one here, are going to lag or follow other sets of regions. And that's another area of intense scrutiny to understand whether or not there's a temporal dynamic that has information in it. It's a good question. So I showed you a very simple way of how resting state correlations are analyzed, but in order to understand large-scale networks, we need to put together many sets of correlations, because a network isn't simply one region connected to another region. It's your collection of regions connected to a collection of the remaining regions. So the way we go about analyzing resting state networks is we start with a set of predefined functionally defined nodes that we believe are biologically plausible. I'm not going to go into the detail of how we generate this, but this is an area we've been heavily invested in as well. So once we have an estimate of where we think those units of the brain are, those areas of the brain, we extract their time course, their bold resting state time course. And then we cross-correlate every time course with every other time course, and you can create a matrix, right? So this matrix just reflects the statistical correlation between a region within any other given region in the brain at rest. And we do that for each subject. And then from there, we can build these graphs and start analyzing graph properties. So doing this, the first things that were done were to understand whether or not young adult brains exhibit any organization at rest beyond what I've showed you in that previous slide. And using this graph theoretic approach, some colleagues of mine in my previous lab published this result demonstrating that resting state brain networks are organized into something we call communities. Communities is a term that comes from the network science of networks. Other people will call them modules. But what this is showing you is sets of areas that are correlated with one another and less correlated with other sets of areas. So each of these dots corresponds to a brain area. And these are this is the average of a group of young adult brains. And what you can see is that there's this clumping, right? There's this modularity. Sets of brain areas seem to be highly correlated with one another and less correlated with other sets of brain areas. And the surprising thing to us that we realized is that this organization actually corresponds to functional organization that we know to exist in the brain. So in blue here is actually areas of the visual system. They're all highly correlated with one another in this graph theoretic framework. In yellow is the frontoparietal control network. You can see the topography on the brain here. And these are all regions involved in executive function. In the red is the default network, which some of you might be familiar with, involved in lots of things ranging from social cognition to autobiographical memory and emotional processing and so forth. But again, this is all at rest, right? And the math doesn't know anything about the function of these areas. The math is just telling us what areas are more correlated with one another. And we can see this beautiful, rich topology that reflects functional organization. So in exploring brain networks over aging, the first thing we wanted to do was ask whether or not brain network organization differs across the adult lifespan, taking this very kind of macroscopic approach of looking at those communities or those systems. And I was lucky to have a great collaborator to work on this, Dr. Denise Park, who's here at UT Dallas. And Denise had collected a study called the Dallas Lifespan Brain Study. And embedded in that study was some resting state scans, which we were able to analyze.
Doctor, we have a question in the chat.
Sure.
It's, does functional connectivity eventually pre-cut structural connectivity?
Does it eventually predict structural connectivity? It does to some extent. So I made a comment earlier that there isn't a one-to-one correspondence. There are functional connections that exist, strong functional connections that exist between regions that aren't directly structurally connected. But you might think of it in the other direction. So structural connections can predict functional connections. But again, it's not a perfect correspondence, telling us that there are unique and distinct sources of information that are available to our understanding.
Thank you.
Sure. So as a part of the Dallas Lifespan Brain Study, over 300 healthy individuals were recruited for participation. And the nice thing was that these participants ranged in age from 20 to 90 plus. And I think this is an important feature of this study and many studies that have been being subsequently collected, because it allows us to understand not just the endpoints of the aging process, comparing a 20-year-old to a 90-year-old, for example, but rather understand the progressive changes that might accompany midlife and so forth. And I think that's really going to be at the heart of understanding why there exists age-related decline. So as I mentioned, included in this data set were measures of brain anatomy, but then also resting state scans. Now, analyzing resting state data, while it's easy to collect, is actually difficult to accomplish, because resting state data, for those of you that are familiar, is actually prone to a lot of sources of non-neuronal variance. Excuse me. So we do a number of steps to minimize those sources of variance in our resting state, but also in our anatomical data. I won't go into the details, but if you're interested in some of the things that we do, which I think are necessary, those are provided in some of these publications here. So as I said, the first question we had was a very basic one. If we look at the general organization in terms of the system organization of the brain that we saw in the young adults, does it exist across adult lifespan? So this is just another depiction of that same pattern of the different brain areas color-coded by their community membership, which again aligns with the functional roles that those regions and those communities play. And for the depictions purposes, I'm going to show you the data set cut into some arbitrary groups of age. So the first group, of course, is 20 to 34-year-olds, but then we also created a group of individuals who were 35 to 49. We're calling them middle early adults. 50 to 64 were middle late adults. Older, earlier age adults were 65 to 79, and 80 plus for older, later adults. And rather than belabor the point here, I'm just going to show you the answer. It seems to be the case that the spatial organization of brain systems is largely consistent, which I think makes a lot of sense. We didn't think there should be massive reorganization of the brain as you grow older, because again, these systems mediate some core cognitive operations, and observing any drastic changes in the membership or in the function of any of the given system would mean something pretty disruptive for cognition. So initially, we thought, okay, things are looking very much the same. But this also allows us to ask much more targeted questions now in terms of network organization. So that's what we started to do. So the other piece of this, which I alluded to, is that if we look at this community structure, again, this is defined by two simple things. The first is a given community or a module of a network is defined by dense within system connections. So communities are highly connected within themselves. The nodes within a community are highly connected. But the other feature that defines a community is that it's sparsely connected to nodes in other communities, or in this case, brain areas of other communities. We could see that here. This area is connected to two areas over here, but in general, the visual system isn't connected to too many other systems, which again defines it as a community. So we refer to this as the segregation of functional brain systems. In a healthy young adult brain, your functional brain systems are segregated from one another, and we think this allows it to function. But as we know from, again, the science of networks, segregation of networks can change. The segregation of network communities can evolve over time. This is an interesting example, especially given the current political climate in the US, whereby the networks here are depicting nodes as senators. And a link between any given two senators reflects voting on similar legislation on at least 100 occasions during the same congressional session. And as you can see, what's being depicted is 3 different congressional sessions up into 2013. And these senators have been color-coded by their party affiliation. Blue as Democrat, red as Republican, and the few independents are color-coded as green. What we can see here is that the segregation of this voting behavior across party lines has progressively changed over time, whereby the different parties are voting on on legislation in a common way less and less frequently. And I think this was summarized really well by The Economist, this line here actually really highlights an interesting aspect of what's being seen here. Though America's political polarization has become a fact of life, it has never been seen so graphically as a diseased brain with few neural pathways between the 2 hemispheres. So I think this is interesting for numerous reasons. And I'm not going to make any comments on the political climate, but I think what this is really showing us is how this property of network segregation can really confer some emergent behaviors of the system that it's characterizing. In this case, the idea is that lots of connections between between party lines is actually a very good thing, right? It shows coming across the party lines. Whereas less connections between them, it might be a bad thing because you have political polarization. And as I said, I'm not going to comment on any of the details here or where I think we are in the current climate. But what I can comment on is whether or not I think it's a good or bad thing for the aging brain. So just to kind of rehash the same idea, having network communities that are segregated but still able to communicate with one another requires a really fine balance of connections, both within and between communities. And I think we can think of it starting from this middle point here. So if you imagine an idealized network with multiple nodes that exist in their distinct communities, going in either direction could actually be a bad thing if you go too far. If you start disconnecting things or disconnecting nodes from their communities or communities from one another and increasing disaggregation, eventually those systems or those communities are no longer communicating with one another, which, as we can imagine, is very disruptive. But going in the other direction could also be really bad, because as you start to increase connections, you start to lose some of the distinctiveness that is conferred by that modular organization. So we think modularity allows these different communities to be functionally specialized. And to the extent that you start adding connections between those functionally specialized sets of regions, you start losing that specialization. So too much communication, we think, is a bad thing too. So just to frame this in terms of brain networks, we think when we look at healthy young adults, we're really in this idealized territory where healthy young adult brains have functionally specialized systems that mediate distinct cognitive operations. And those systems are connected to other sets of systems, thus allowing the brain to operate as an integrated whole. But then the question is, does aging impact community or system segregation? It's possible that increasing age is associated with increasing disconnection of the communities, and we're going to go in this direction. Or maybe you'll see greater connections between systems, and we can go in this direction. But allowing the science of networks to inform this discussion gives us a framework to think about what aging might be doing to the brain. So what I'm plotting here is, again, data from the Dallas Lifespan Brain Study. But in this case, I'm plotting each individual subject. And what we did is we simply split their connections between all the brain areas with all the other brain areas. And we just split them according to whether or not the connections were within systems or between systems. As you can see here in this top line, when you look at the individuals, the magnitude of their within system connections seems to decrease with increasing age. So connections amongst the regions in red, or the default system, are decreasing with increasing age. And we thought this was going to be a general feature of all connections. But the really surprising thing, and you might notice it here, is that when we looked at the other type of connection, or the between system connections that are spanning the multiple communities, they actually increased. And we were surprised by that. Because this suggests that the nature of the connections is actually differing in different ways as one grows older. We summarized this basic property in a measure which we called system segregation, and that's described here in this box. But what you can see is when the point of this measure is to summarize the extent to which those different communities are separate from one another versus together. High segregation, again, means your communities are highly separate from one another, and lower segregation means they're clumping together more and more. And what we can see is that with increasing age, your system segregation it seems to be decreasing progressively. So older adults have decreased segregation relative to relative to younger adults. This is another depiction of that same property. Again, in this case, I've just divided the subjects into 4 age groups. And if you stare at this long enough, you'll probably see the same things that we focus on. I can try to highlight them here. In this case, I've got 4 groups of younger adults, early, middle, late, and older. And if we look at the arrow, we could see the within system connections decreasing with increasing age. This is, again, is a visual system. You can see it's highly integrated here in the young adults. And as you grow older, it's starting to fan more and more out, which reflects the fact that your within system connections are decreasing. Conversely, if we look at the circle here, right, this is highlighting control systems of the brain, or association systems of the brain. There are sparse connections between systems, but as individuals grow older, those between system connections start to increase such that there's a lot more interactions between the different color groups, and there's a lot more splitting up of different things that should be holding together.
A question.
Yes.
Is there a specific clinical manifestation? I know this is kind of maybe complicated for that, but is there like a small example of that undifferentiation being manifested? Like,
in terms of behavior?
Yes.
Yeah. So again, this is all in the health, quote unquote healthy and typical kind of range of cognitive function. But I'm going to show you an impact of this in a moment. Actually, I'm going to show you that
Nice.
In a few moments.
Cool.
The first thing I'll address is whether or not this is just a one-off example that we published that didn't hold up, because that's a critical question. If this is going to have any cachet and any value towards us understanding aging, it has to be a reliable finding. So how reliable is the relationship between brain system segregation and adult age? So again, on the left here is that same figure we published, but just to summarize, the age versus system segregation relationship, it continues to be observed across multiple studies that are all using variants of similar methods, but are again all demonstrating that this pattern in distinct subject groups and in distinct populations holds. With increasing age, we see decreasing segregation. Now, in our own hands too, we've been seeing this time and time again. This seems to be a really robust effect. We have multiple other studies on the go, some of which involve aging, but some of them which do not involve aging, but they might have an age component. And I can show you a summary of some of those observations. So again, this is data now coming from our own lab, processing the data using our own methods. This is data from Washington University, the ADRC there. And we're working on this data set for another project. But again, there was a major age component where the subjects came from different age from different age time points. And there's a very strong effect of system segregation as a function of age. You might be familiar with the Human Connectome Project, an effort to collect data on a large group of healthy young adults. It's got developmental and aging components to it now. This is the first effort that was conducted, whereby data collection was done on individuals from 20 to 36. And in this case, there's a lot of data per individual. Even though there's a truncated age range, we're a little bit surprised by this. We still see this pattern. Even within the 20s and early 30s, increasing age is associated with decreasing segregation. This is another data set that we've been working on. The focus here has been on major depressive disorder. The goal of that study is distinct from one of looking at segregation, but here too we can see that independent of whether or not and which circuits exactly depression is modifying, what we see is amongst the healthy controls and among the patients, you see this strong relationship between age and system segregation. And finally, just to drive home the point, this is another data set done with Francesca Philby at UTD, where she was interested in understanding chronic marijuana users, again, for a different purpose, but again, there was an age component, and there too we see amongst controls and amongst users, there's this robust relationship. So is a system segregation a reliable measure of an individual's brain network organization? You can imagine I can describe these things across individuals, but what if I take the same individual and bring him in to scan them and then bring them back on a subsequent day? Is the measure going to be roughly the same? We haven't done anything to fine tune the measures or the measurement or the data acquisition, but what we do have access to is a set of subjects, again, from the Human Connectome Project that were scanned on 2 separate days. And you can see with enough clean data, the measure is fairly reliable. It's not perfect, but again, we haven't done anything to really fine tune the measurement of this. But what we're seeing is that a given individual, if they're high on one day, they're going to be high on another day, thus giving us some hope that this measure might have some value. Now, this question was asked about whether or not there's any clinical indicators or clinical manifestations. And of course, that's a critical question for us. And we're trying to
question for the last slide.
Yes.
Okay, and theoretically, if you have this imagine, you expect that this line should be in the diagonal from 0.6 to 0.8.
That's right.
So that means that there is some problem with the reliability in the second time?
Yeah, so it's again, as I said, I'm not, well, I think, so it's not at unity, but I think this is an outlier right here, right? If we didn't look at this outlier, it actually probably would fit something along the diagonal a bit better. I haven't done anything here to understand the outliers or how they're contributing to the estimates. But yeah, I think in an ideal scenario, they would be at unity, and any individual who's off unity, like these people are certainly off unity. The question is what's happening, what's differing between their two sessions. It's not systematically above the line or below the line. This just could be measurement noise. It could be quality of the data that varies as a function of participants. We haven't figured out those details yet, but those are going to be really important to figure out as we move forward.
Okay, thank you.
Thanks. So does an individual's brain system segregation relate to his or her cognitive ability? As a part of the Dallas Lifespan Brain Study, there was measures of cognition that were collected outside of the scanner. And we were able to take those measures and identify that they were composed of 3 principal factors. And one of those factors was actually a factor that related to their episodic memory ability. So there's multiple tasks that contribute to that factor. So what I'm going to show you is a relationship between system segregation and long-term memory, long-term episodic memory. And critically, we've regressed out age from both these measures. As I've just shown you, age relates to system segregation, but I also showed you earlier that increasing age is associated with poor long-term memory. So we've accounted for age. So what does it look like? Is greater segregation associated with better episodic memory or long-term memory, or worse long-term memory? I think people had bets in either direction when we started presenting this work. And it turns out that people with less segregated brain systems tend to have poorer long-term memory ability. So again, individuals on this upper quadrant here have greater segregation of their systems, and they also have better long-term memory, whereas individuals in this lower quadrant have lower segregation and lower long-term memory. And just to restate what I mentioned earlier, this is after accounting for age-related variance. And that's further highlighted by the color coding of these dots. So what you can see is that there are individuals who are older who actually have relatively high system segregation, and their memory, their corresponding memory is actually quite good. Conversely, we also have individuals who are younger who have lower system segregation, and their long-term memory ability is actually quite poor. So again, this gives us some measure or some indication that there is a reliable, or there is a meaningful cognitive construct or manifestation of what we're seeing in terms of brain network organization. So does that answer the question that was posed regarding any indicators? Okay, I don't think there's, okay. So I'll move forward. So just to wrap up, I'll close with a final chapter in one of the things that we're pursuing. And that relates to the other question that I started this talk with, as to whether or not there are any modifiers of system segregation across the adult lifespan. Or another way to phrase that, which is more in line with what I mentioned, was whether or not individuals who are more vulnerable to age-related decline exhibit differences in brain network organization. Again, if this is going to have any value towards us understanding brain function and brain decline and aging, then we should be able to, this should align with some of the other indicators in terms of vulnerability to age-related decline. So we've been focusing many of our efforts on environmental determinants of age-related decline and disparities in environments. Socioeconomic status is a crude construct, which can be defined by education or income or occupation. But the reason it's important to think about is because it often defines our access to resources. And those include our access to nutrition and adequate healthcare, but also access to cognitive stimulation and maybe as a more global issue, levels of stress. And socioeconomic status has been an area, has been a variable of interest because lower SES is associated with worse cognition and greater risk of Alzheimer's disease. So thinking about in disparities in SES or in disparities in the environment or differences in socioeconomic status is important in order to understand what might be some of the causes of some of the age-related illnesses. Just to place this into some broader context, if we think about SES as an important variable, and we look at low-income and middle-income countries as opposed to high-income countries, the growth in the number of people with dementia is actually greatest for low SES countries. Again, telling us that there must be some environmental modifiers of age-related decline. And it's critical to understand those modifiers if we're going to slow down or eliminate any of those age-related declines or illnesses. Finally, I think this is another really poignant data point to consider. Developing nations are experiencing a rapid increase in the number and percentage of older individuals. So what's being shown here is the time required or expected for a given percentage of the population age 65 and over to rise from 7% to 14%. So whereas countries such as the United States and France and Sweden took a considerable amount of time for the population to rise to that level, the proportion of the population to rise to 65, to rise to 14%, that progression is much more rapid for developing nations. And again, I think this is another important comment that was made. It seems to be the case that some countries might be growing old before they grow rich. So again, in order to understand, I think it's really important that we understand how environmental disparities are going to impact brain aging if we're going to make any progress in alleviating age-related illness at a global level.
Dr. Rui?
Yes.
We have two comments, two questions.
Mm-hmm.
One asking about, is this segregation is only related to memory, or what about other cognitive domains or even behaviors such as task management? And the other question is, could you comment on basically on sex differences, particularly in relationship to women after menopause?
Yeah, great questions. So the first question about cognitive ability, whether or not there's some specificity in terms of the relationship between segregation and memory function. So in that first study, again, we had three constructs. One was memory, one was executive ability, and one was verbal ability. And the only one that exhibited a relationship for us was long-term memory ability. Now, other groups have found there's subsets of systems that exhibit this pattern, and those are uniquely related to other domains of cognition. And I think that's probably where the story is going to end up. We're examining different specific systems that are mediating specific cognitive operations will probably relate to specific domains. I think another challenge for us, again, is the lack of, or maybe an imperfection in the way we characterize cognition. These constructs are built from sets of tasks, some of which are less perfect than others. So I wouldn't put too much weight on the fact that it didn't relate to the other domains. I would think rather that maybe we just haven't characterized those domains as adequately as we should be doing, and that's something we're trying to fix now. In terms of sex differences and pre versus postmenopausal women, we haven't seen any significant impact of sex as a categorical variable. I think hormonal change, though, is going to play a significant role. And in a follow-up study, which I'll briefly describe at the end, we're going to start characterizing those important mediators of these relationships.
Thank you.
Sure. So just to show you where we're going with this, while we don't have population, global population level data, we could start asking whether or not there are any environmental determinants or modifiers of some of the patterns that we're seeing. And again, the way we've done that is just by leveraging the data set that we have available to us here. And this is a broader segment of this data set because additional subjects were recruited after that first study that was reported, and they came from the Dallas-Fort Worth area, and they did included in this is a relatively broad representation of education and of occupational status. So we use those as combined measures to define SES. So we took an individual and we defined their socioeconomic status according to their years of education completed and their occupational status, which is just a coding of their occupation into a standardized measure. What we did then is look at those system segregation as a function of this SES construct. And while we do this at in a continuous fashion, I'm going to plot it in a way that just creates categories. The first category is, again, an age category. So I just have 4 age groups, as you've seen before. But the other thing I'll do is I'll just depict it in a way where I'm going to split higher SES and lower SES just using a median split. So just simply taking the median of socioeconomic status and showing you higher versus lower. Just as a very crude depiction, but to drive home the point that I hope you'll appreciate. So this is higher socioeconomic status as a function of age group, right? And we could see that pattern of decreasing segregation, which is most prominent from 35 onwards in this way of graphing the results. But what happens with lower SES? This is what we see. Lower socioeconomic status is associated with reduced system segregation, but we're seeing it primarily in middle-aged adulthood. Okay, so whereas the younger adults don't seem to show a big separation in terms of their SES and their segregation as a function of their segregation as a function of their SES, the middle-aged adults do show that those differences where higher SES is associated with higher system segregation. Surprisingly, the older adults didn't show that the effect that we anticipated, and we think this has more to do with issues related to participant recruitment than the actual observation. For those of you familiar with some of the work in aging, you know, there's problems of selection biases and attrition biases where individuals that are less healthy are less likely to be recruited, and I think that might be playing a role in this. So the lower SES individuals we have, I think, are simply very healthy relative to some of the other age groups. Now, speaking to that, this relationship persists when we control for a number of critical factors, which I think are important to consider. And those include their demographics, so their self-reported race and their sex, but also the available measures we had of physical health, of mental health, of cognitive ability, since I've already showed you cognitive ability predicts system segregation, but also, quite importantly, their childhood SES. As you know, as you likely know, the relationship between SES and childhood development is pretty robust. So it's important to us to establish that any observed relationships aren't just a consequence of differences in childhood SES that have been propagated forward. So here we've used parental education. When you have an adult individual and we control for their parental education and just consider their own SES defined by their education and occupational status, we seem to see this separation. So this is a summary of some of the observations that I've been describing to you. The first is that the segregation of brain systems seems to decrease with increasing age in adulthood. And I've shown you a few different ways of looking at that and ways to think about that. Reduced system segregation is associated with poor cognitive ability. In this case, we've been focusing on episodic memory. And finally, at least in middle-aged adults, lower socioeconomic status is associated with lower system segregation. And we think this suggests that there exist environmental factors that impact brain network function and vulnerability. So what are we doing now? Just to close up, I'll just give you a sense of the direction we're taking this work. The first thing we're doing is asking whether or not brain network segregation might be useful as a measure of brain health. All the work I've shown you thus far has been cross-sectional in nature. So we've looked at different individuals that come from different ages, but we're now bringing together multiple large longitudinal data sets to start to ask whether or not we can see brain networks changing as an individual grows older, in both health, but also in their transitions to disease. A related aspect of this is asking whether or not that network organization might be modified by changes in brain degeneration. I think some of the differences in segregation we see and some of the changes that we're seeing are a consequence of progressive degeneration. Finally, the million-dollar question is whether or not we might be able to predict who might be vulnerable to brain disorders based on their network patterns prior to any clinical manifestation If we're able to do that using this measure or this measure in combination with other measures, I think we've come a long way in slowing down the march of age-related illnesses. Another study that that we're focusing on actually focuses on the last study I presented. So as I mentioned, the socioeconomic status construct is crude, right? But we think it reflects some underlying features of an individual's environment. So what we need to do is understand what is it about SES or the individual's environment that is moderating their brain network organization. And in a new study that we're about to start collecting, we're going to be asking just that. So the design of the study is to include representative sampling of lower to middle-class, middle-aged adult participants from the DFW area, and then do longitudinal assessment of their changes in brain network organization and their cognition. And the critical element is that we're going to do extensive characterization of their changes in health and lifestyle, including stimulation, socialization, exercise, diet, their communities, sleep patterns, and then other measures of stress and health and well-being. And this study will start once we feel it's safe to start collecting the data in the context of the COVID-19 pandemic. So that's all I have to present for you. But before I end, however, I do want to acknowledge the people that were instrumental in making this work happen. And those are, are particularly, I want to focus on members of my lab, including Kyla Chan and Liang Han, whose work I presented today. But there are other members of my lab who are all involved in this work, who motivate much of what we do. I also want to highlight members of the UT Dallas community, including in the School of Arts and Technology, members of UT Southwestern Medical Center and my collaborators there, and then members of the departments of neurology and radiology at WashU School of Medicine, where I conducted my postdoctoral training and with whom I still collaborate. That's all I have for you. Thank you for your time, and I'm happy to take any questions.
Thank you very much. Dr. Wig, this is really fascinating. So let's open the floor for questions from our audience.
Hello, Dr. Wig. This is Kathy Chunsu. Very informative talk. I learned a lot. I have two questions. One question, I'm wondering if there's a genetics or lifestyle or different ethnic population also impact on cognitive function as modifiers.
So, sorry, the question was whether or not
genetics or a different ethnic population. Being individual, we have different genetic profile, and we are different ethnic group. So this can be have impact on cognitive function when we're getting old.
Yeah, so I can turn my camera on. There we go. Hi everybody. We haven't looked too much at genetic profiles yet, quite simply because we don't have the data available to us to start to do that. What we have done is looked at APOE status, which is a genetic risk marker for Alzheimer's disease, given our interest in memory disorders and our interest and the observations that related network effects to memory function. And I can tell you there, we haven't seen any robust interactions between APOE status, which is again, a risk factor for Alzheimer's, and these measures. Yeah, that's all I can speak to in terms of potential genetic differences at this point.
Okay, thank you very much. Second question, do you think if we increase physical mentally activity will improve brain network and improve cognitive function for the aging population? I mean, for the seniors.
Yeah, that's a great
physical mentally activity.
Yeah, that's another great question. I'm involved with another interventional study where we were trying to look at whether or not learning a new task, specifically digital photography, would modify networks, and that work is still ongoing. I think there's multiple groups now that are interested in this, whether or not, first of all, whether or not cognitive training works at a cognitive level, right? Does it improve cognitive function? But then whether or not there's any brain changes that accompany those cognitive changes. So I think the answer there is still outstanding. I don't think it's going to hurt you though. So I think if the question is whether or not we should be engaging in intellectually and mentally stimulating activities, I would say yes.
Thank you. Thank you.
Dr. Wig, I'm wondering what network segregation really means in terms of cognitive ability. Does it mean that the brain neuron functions are more specialized or more independent of each other? What does segregation really mean? I'm not very clear. Does it mean that when one does a mental work, fewer areas of the brain are involved if the segregation is high, and more areas of the brain are involved if the segregation level is low?
Yeah, great question. So yeah, what I think this, what this is depicting is a basic property of brain function, whereby distinct sets of brain regions are functionally specialized and permit specific sets of cognitive operations, right? And we could talk about that at the level of neurons, circuits, or areas, or systems. So in order to have an efficient and effective systems or networks, at least in terms of the brain, but also in various other systems, you need modular organization, right? Because that permits specialization. And this measure of segregation is just a statistical way of quantifying the extent to which you have that specialization via patterns of resting state correlations.
So as we grow older, this specialization level declines.
That's right. And we've seen, so I didn't present the work, but we're seeing this in terms, again, this is all at rest, right? But when we look at task activity, we have data on individuals doing tasks. And we could see that individuals who have less segregated networks, their task activity is less specialized. So regions that are otherwise performing a given task are now performing it with other regions that shouldn't be performing that task.
I see. Thank you.
Sure.
So basically, it's a way of looking at plasticity of the brain.
That's right. That's one way of thinking about this. Looking at what changes, what brain changes are accompanying increasing age.
Actually, I want to jump in there. Great talk, by the way. Really enjoyed it. Learned a lot. And and I was also going to ask about if you could maybe entertain the idea of linking it to plasticity, because I know in some individuals, right, the changes in the brain, the plasticity that takes I mean, it's not always a negative thing. It can be very positive. And as we age, clearly we use maybe different resources, different areas to solve a problem. So are you able to kind of talk about plasticity that might potentially be good plasticity, in the context of somebody who grows older and the plasticity is good versus somebody who maybe, I don't know, or plasticity that might be more negative? I don't know if you can tease that out or talk about that.
Yeah, that's another great question. Again, I alluded to this earlier when we started looking at the cognitive data. I think a lot of people were split, right? Because as we know, as we grow older, we get, one idea is that people get wiser, which I think is a fair statement to make. And we see that in terms of world knowledge increasing. So some of the changes are certainly going to be to the benefit of the individual. We haven't been able to dissect yet the specific patterns that might be mediating the beneficial things that accompany aging versus the detrimental things, right? Right now we're taking a kind of a more bird's eye view of like large scale network organization. But I think there are certainly going to be some of these patterns that reflect beneficial outcomes. We just haven't drilled down into those patterns yet.
Okay. And another quick follow-up. I know that among our group, I know that there's a number of people that are interested in resilience. And I'm just wondering if in your field, people are looking at it, well, maybe networks that maybe are associated with resilience. Because I think most people probably don't talk about that, but that would also be interesting, along the lines of the positive plasticity, but resilience. I'm just curious if that's something that's starting to appear in your area.
So, the idea of resilience actually comes from the science of networks, right? So resilient networks are networks that are able to sustain targeted damage, yet still maintain function or adapt very quickly, right? So we think about faulty power lines, right? But yet you still have transmission of electrical currents. Those are typically resilient electrical networks. So that is the idea of resilience is actually a core concept that we're interested in exploring these things, but again, using the framework of the science of networks.
And would you say if we're searching for it, like resilient networks, or what search phrase would you use? I'm just curious, like a PubMed search, just resilient networks? Or?
Yeah, resilient networks.
Okay.
You'll get good hits.
All right. Thank you. Appreciate it.
Sure. Thank you for the questions.
It seems that the high level of segregation is correlated with, more efficient cognitive information processing. So could that imply that people with higher intelligence have a brain with higher level of functional segregation than people of lower intelligence?
I think the intelligence construct is challenging as well to grapple with. I think, we haven't looked at G per se, but our episodic memory construct will be related to G to some extent. But I think there is other work that's trying to relate measures of intelligence to measures of network segregation. I'm just not familiar with whether or not they've come to any conclusions on that work yet, though. But at least in terms of cognitive ability and specific domains, we are seeing relationships. And our group's not the only one now. There's been, I forgot to mention, there are other groups that are starting to see relationships between network segregation and executive function when focused in on specific systems. But whether or not there will be some direct relationships between general intelligence and any of these network measures is unknown, or is presently uncertain.
Okay.
So if this is a matter of function and not structural, so there is hope that with neuromodulation we can have younger brains. I mean, in terms of the networks.
Yeah, I mean, you know, that's the optimistic perspective on this, right? That you were able to modify. We know you're able to modify your resting state networks with extensive changes in behavior. So, you know, like a pipe dream would be to identify the specific factors or the sets of things that you need to do in order to modify these functional relationships, despite degradation in the anatomy, which might be somewhat inevitable. But, you know, I think my suspicion is that a lot of the things that we know to be good for your general health and for your vascular health will probably be good for your brain health.
There is a follow-up question on an earlier question in the chat. Have you looked at retired versus still employed elders in terms of brain function segregation? Also, have you heard the elders who solve puzzles, crossword puzzles, etc., keep better brain functions? Is any analysis on actually putting problem solving or puzzles?
So yeah, we didn't. So we did have retirees in the population that we examined. And those primarily came from the older cohorts. I don't think there were, I mean, because we didn't see any separation as a function of socioeconomic status, we you know, further dividing them according to retirement status was actually further complicating the analysis. So we haven't seen anything specifically there yet. But I think that speaks to the follow-up, which also is really, you know, whether or not mental stimulation is going to modify these networks. As I mentioned, we have something ongoing that's looking at that specifically, but I can't speak to whether or not I think, you know, a specific thing or set of things that you do is going to change your networks. We don't have evidence for that yet.
Is this segregation, as far as you know, human, or it also happens in primates?
Yeah, great question. So modular brain organization is a property of across species, right? It's just a general principle that we see across rodents and non-human primates. So that those are definitely amenable to targeted inquiry of these kinds of questions, and whether or not there are environmental or other factors that can be actually modified, that would actually modify the measures.
Dr. Wick, this may be something of interest to you. I remember I read something in a cognitive psychology textbook that says that when you know, they compare more intelligent people with less intelligent people, they have these two groups of people do the same mental task. The more intelligent group shows more, you know, specific areas in the brain highly activated, whereas the less intelligent group shows a more diffuse, the general area in the brain that is activated. So that, you know, those two patterns seem to be the same as, you know, more segregated versus less segregated.
Yeah, that's really interesting. I'm not familiar with that observation, but that would fall in line with actually the prediction that you made earlier.
Well, wonderful. I just have a last question on my side. It has to do with blood pressure. I'm very interested in blood pressure. And we know blood pressure affects the brain, and it actually is a modifier of brain structure. Have you looked at brain function?
So we've controlled for blood pressure as a part of our physical health construct when we were doing that socioeconomic status work. We do have some data on some data from these individuals using the cardiovascular, using respiratory challenges. So it's in the pipeline as to whether or not there are going to be differences in neurovascular coupling that ultimately related to blood pressure or any other cardiovascular indicators. But there's been work from other labs on this, and those haven't shown any robust relationships between those measures and the network measures yet. But that's something we hope to pursue soon.
Great. Thank you so much. I don't know if any other any participant has a comment or question. I think you've given us a lot to think. I think we are using the whole brain.
One more question. Can you think of network segregation as synonymous with executive control function?
No, I think the former is reflecting the architecture of the network, and the latter is a process. So the operations of that network, and that's mediated by specific subsets of the network.
Okay. Very interesting.
Thank you very much.
Well, thank you. Thanks everyone for coming.
Thank you.
How does the pandemics affected your work? I saw that you mentioned in one slide that the recruitment, I guess, or the phenotyping is being affected.
Yeah, so I mean, my lab has two arms, right? One of those arms is the analysis of data sets that have already been collected, both from members of our group, but also our collaborators. And the other arm is data collection. So we've had to shift our focus on the former because we're not going to collect any new data given the prevalence of the pandemic in the Texas area and in Dallas-Fort Worth. So we were going to begin collecting data in March, and then of course, the world decided otherwise. So we've been in a holding pattern. It's been an opportunity to perfect the design and perfect the measures, but we're waiting to start collecting.
Yeah, well, we appreciate very much that you took the time to talk to us during this time. It's hard. But remember, we have a community here. We meet every Tuesday. So I'm glad to see other speakers have come, join, and we all are in this together, as it is true. And thank you so much for coming, and we really appreciate you taking the time.
Well, thank you for bringing together everybody, and this is a wonderful forum, so I'm excited to interact with you all more.
Thank you. So we have few announcements.